Okay, hello and welcome to the Global COVID Lab meeting. My name is Sabrina Welsh and I'm the Director of Programs and Operations at the Human Vaccines Project, and I'll be your moderator for this webinar. I wanted to start today by thanking all of you for joining us and taking the time to participate in the lab meeting during this challenging time. While the scientific community continues to press forward for a vaccine, we want to acknowledge that science is part of society and we have a role to play in creating equal access to science and health interventions. We at the Human Vaccines Project stand in solidarity with the vulnerable and historically oppressed groups, both here in the US and globally. They have borne the brunt of this pandemic and are concurrently experiencing institutionalized racism that continues to threaten lives. As difficult and uncertain as the past few weeks have been, this is an opportunity for us to work together to tackle these issues and improve circumstances and health for all. To meet the goals of ending the pandemic, we must continue to work for a vaccine that is safe and accessible for everyone. Today, we are gathered here to take up the topic of novel approaches to a COVID-19 vaccine and aim to bring the latest information from experts across the field in a forum open to scientists globally. To date, more than 1,100 people have registered for the Global COVID Lab meeting, and we've received a lot of great feedback from attendees from the past few sessions. As new and critical COVID-19 data are released each week, we aim to bring you the latest information from experts in the field, and we're really pleased that so many of you are joining us for these discussions. A quick note before we begin. The information presented today contains some pre-published data and is currently under peer review. So at the request of the author, we ask that you do not reproduce or disseminate the data presented in any way. This session will be recorded and available on our website and social media channels for you to review. And we are very excited today for today's speaker, Dr. David Weiner. Dr. Weiner is the Executive Vice President of the Wistar Institute and Director of the Vaccine and Immunotherapy Center. He is also a professor of the Wistar Institute of Immunology, Microenvironment, and Metastasis Program, and a W.W. Smith Charitable Trust Professor in Cancer Research. Dr. Weiner directs a translational molecular immunology research team focused on creating novel approaches for disease prevention and treatment using synthetic nucleic acid technology. With help from collaborators, his group is one of the pioneering research teams in the field of DNA vaccines and immunotherapies and advanced the first studies of DNA vaccines. The Weiner Lab, with collaborators, has contributed to synthetic DNA platform advances through the first clinical trials of a Zika vaccine, an advanced MERS coronavirus vaccine, and developing a second-generation Ebola vaccine, among others. In oncology, the Weiner Lab has helped to advance immunotherapy approaches for HPV-associated cancers, prostate cancer, and glioblastoma multiforme, which are currently in clinical testing. His work has led to the first clinically efficacious therapeutic DNA vaccine outcome, in this case for HPV cervical intraepithelial neoplasia, which is now in licensure trials. The Weiner Lab has published over 400 papers, chapters, and reviews, and Dr. Weiner is an avid supporter of the training and education of young scientists. In today's forum, he will describe some of the recent advances in the synthetic DNA vaccine approach, of particular importance for emerging infectious disease outbreaks, including COVID-19. During the presentation, all attendees will have the opportunity to send questions about the data presented today using the Q&A function in Zoom. I'll collect your questions and ask Dr. Weiner a broad selection of these questions after he's finished his presentation. We'll have about 25 minutes for discussions, and we really hope you'll take the opportunity to ask Dr. Weiner your questions about the data presented today. And make sure you use the Q&A function and not so much the chat so we can collect them all in the same place so I don't lose track of your questions. Thanks. It is now my pleasure to introduce Dr. Weiner from the Wistar Institute.
Thank you, Sabrina, and thank you so much for the important message. And it is really an honor for, I think, all of us who participate in this challenge of developing a safe and effective vaccine for global use, that we are all together, joined together, fighting the virus to protect all individuals. So I have several disclosures to start out with. I work with several different companies, and they're listed here.
Oh, go ahead and share your screen, Dr. Weiner.
It's not being shared right now.
We don't see it quite yet.
No, I don't see the share.
It should be at the bottom of the Zoom panel.
I no longer see the Zoom panel. Oh, there we go. Got it. We're back.
All right, great.
There
Okay, super.
All right, so these are my disclosures, which are monitored by the Wistar General Counsel's Office. So, I mean, this is a wonderful forum put together by the Human Vaccine Initiative, and we really appreciate it. And of course, we're joined together because of these early reports from China originally of a outbreak of a new coronavirus. This is the third time in this century. As 20 years, we've had coronavirus outbreaks. The first SARS, of course, MERS a few years ago, and now COVID-2. And it was described originally as disease with severe lung involvement, and now we know that's due to, in part, the ACE2 receptor, which is its target. And this resulted in an explosion of infections, first locally and then disseminated around the globe in just months. And particularly disturbing with this infection is the high percentage of people who wind up with severe cases. We have about 80% of individuals who have less significant cases or mild pneumonia, which is not really that insignificant. And then we have close to 20% of cases, somewhere between 16 and 20%, that can be quite severe with ICU visits and progression of about 20% to a quarter of those to acute respiratory distress syndrome, quite a serious outcome with a high, significantly high mortality rate. And and this disease, while it is less taxing to young people, does make many young people pretty sick. It does have a lower mortality rate. It also is a relatively long illness, as shown on the upper right-hand panel here, with a several-week time course frequently, including a relatively variable upfront incubation period. So just a few months ago, when we were still debating the impact of this infection and its spread, a report by Ferguson et al. Described really the impact that it was going to have, focusing in this particular case on England and the United States. And this report was kind of a wake-up call for several institutions that this was going to be a very serious outbreak that was not going to be controlled simply, and that we needed both risk mitigation programs such as social distancing, case isolation, et cetera, but that until we had a vaccine, as I put the arrow, it was really not going to go away. And we were not going to be able to resume sort of the way we were before. And clearly this disease has impacted every aspect of our lives. I do want to make a caveat, though. This paper did not really model therapeutics, and I think we all believe therapeutics could really change our opinion of some of the impact of this disease. And so we look forward to those developments as well, which are moving along quite rapidly. And this is a picture in the US, including our risk mitigation efforts. As outlined, we have flattened the curve, and we do seem to be decreasing cases, but it's going to take a while. And we're hopeful about more separation in the summer months. But I think the need for vaccines and therapeutics has never been greater, remains extremely high. And these are a few collections of where we are, almost up to date with this outbreak. And in the upper left-hand corner, I have a slide that was originally presented by Rena Ropoli's group, talking about how outbreaks have been going for the most part, and usually public health policies and other social controls and medical interventions can really impact them quite rapidly. But it's clear if you look at the graph on the right, which comes from the Johns Hopkins website, globally, we're not in that situation at all with this outbreak. And the outbreak is moving. The United States just a few weeks ago was in the lead. We still are the highest number of cases, but now we're seeing an outbreak, very large outbreak of cases in South America, including Brazil. And the global cases are over 6.5 million, and the US cases are over 1.8 million, with a very high mortality rate of debated somewhat based on the true case number of as low as 0.1, but as high as very few percent. And there's been a lot of discussion about herd immunity and natural herd immunity, as well as vaccine-induced herd immunity. And this is just a cartoon. It was made by a graduate student in my lab, Zhang, who really was trying to model, give an example of the impact of herd immunity. And of course, we know that this has had a tremendous effect with our licensed vaccines, but through natural infection, it would take quite a bit. Philly now, the estimated seropositivity rate, which is still debated, is around 10%. So without a vaccine, we would have an enormous way to go. We think about measles, for example, greater than 90% herd immunity is required to stop this disease. Now we think about delivery of vaccines, which is kind of on the next slide, and I'm focusing on vaccines that are actually produced within the person we deliver them to. We have our traditional viral vector systems, which are some of our hallmark vaccines, and so these kind of vaccines really have two properties. The ones that are produced inside, they can make antibodies, of course, and also, though, they can be associated with generation of CTLs, and that's actually a vector-dependent phenomenon, how well viral vectors produce CTLs, and I believe that's also going to be true for the nucleic acid approaches, and we still need more work in that area. And basically all of them need a delivery system and a delivery approach. So viral vectors, of course, have a virus particle with that needs an attachment, and that is basically how it would, even from a local injection, might spread through the body and get in different places and produce its local produce its immune response, generate its immune response from the antigen produced at those local tissues. RNA similarly has a delivery system, which is the nanoparticle, which protects it and allows it to travel to sites for its expression. And the RNA tends to follow the targeting of the particles. Synthetic DNA is different in that different in that targeting sense, because we use now the infectious diseases is ID delivery, a local small 3 millimeter site delivery, and that's all local. And so the area that's transfected in the area of antigen expression is entirely local, not at distal sites. So there's different qualities of these type of approaches. They have different phenotypes and advantages, and we're going to learn a lot more about them during this outbreak as we see data from different groups. I'm going to, of course, focus on the DNA side, and because of the simplicity of these devices and the better formulations and delivery, it's a very reproducible approach. There is, of course, no there's no integration. They're temperature stable formulations that we'll talk about more. And interestingly and importantly, they can handle very large inserts, as insert size is not a problem. And of course, because it's nucleic acid, it bypasses vector serology issues, allowing repeat distribution and distribution in without concern of previous exposure in populations. And this is a slide from Jeff Ulmer showing sort of an argument he sort of constructed around how fast one can make a DNA, an RNA. And of course, RNA has started with a DNA that is the source of RNA transcription. And so I do want to point out that DNA, of course, stands in that category as being quite rapid as well, of course, and you don't have to, these sides, it actually has a different developmental pathway. And it's a different pathway, and so gives a different option for production, et cetera. And I think it's interesting that the RNA approach and the DNA approach were two of the first approaches that were advanced in the United States. Both funded by CEPI, which we'll talk about later. And so this is kind of a summary of some features of the delivery devices. This is a cartoon of it in the upper left-hand corner. And the combination of high concentration formulations, highly engineered inserts, smart devices that control the energy delivered to give equal delivery, which is extremely important in this, and basically improved, has dramatically improved the consistency. And now we're talking about DNA in multiple cases in the clinic for different diseases for generating antibodies and CTLs. I'll talk about that. And also we've published quite a bit on experimental approaches of using them for immune therapy, which I will not be addressing in today's talk. So Kate Broderick lent me this slide from Inovio, and these are some of the collective features that have come out in multiple publications from different members of the collaborative team. Basically, in humans, we've reported T and B cells. I'm going to give you an example of that. You can put, of course, simple to formulate multiple antigens. There's not a competition, and you can give different doses by the mixing of those antigens. It's non-replicating, non-integrating. I talked about that. These devices are quite small, as shown here for skin delivery, comparing it to a cell phone. One of the prototype that was used to lead to this now field use one, room temperature storage for a year, rapid, scalable, and manufacturing. And this is an example of this type of device compared to quite common manufacturing devices that are very simple and able to make millions and millions of in a very rapid fashion. So this is an example of a similar type of device manufacturing pipeline. So I'm going to give some examples that focus on some of the highlights we've seen in the clinics. I'm going to start with cervical therapy, immune therapy, and for generation of CTLs, and it highlights several features. This is where we delivered 4 different oncogenes, of course, bladed in their oncogenic function as a single mixture of DNA cassettes for HPV immune therapy of high-grade cervical intraepithelial neoplasia, and that disease pathology sections are shown here in the right-hand side. And what you're looking at is a clinical trial of women, just under 200 women, vaccinated either at vaccinated 3 times at week 0, 4 and 12, and then examined at week 36 by biopsy for effect on the lesion. And so we're vaccinating in the arm, and we're looking for CTLs that would go to the lesion in the cervix and clear the lesion. And so this is the placebo. There's no clearance, obviously, and here's an example: pre-post, pre-post, pre-post clearance. And also these women not only go histologically negative, and they also, their cervix clears visually, and they go PCR negative, a large number of them. Overall, 50% of the women get better. They regress, 40% regress to normal and clear. And here's another example of, and these are double-blind studies with a pathology adjudicated panel showing another woman's cervical biopsy pre and post. And on this side, you're seeing the matched biopsies. And what you see here is infiltration of CD8 T cells at the week 36 time point that were not there at time 0, again, supporting the induction of CTLs. And this shows how long responses can last. This is following their seroconversion out 35 weeks, for example. And they can last for, certainly we have data out between 1 and 2 years now. Here's a more aggressive example. This is a study done at UPenn led by Chara Agarwal in collaboration with AstraZeneca and Inovio, where we're looking at driving head and neck cancer CTLs, again, from peripheral vaccination. You're looking at cold tumors on the top, and you can see they turn hot, which means they turn brown from CD8 T cells invading the tumors in just a few weeks. And so basically pre and post, you can see enormous amounts of CD8 T cell invasion. Overall, 90% develop from cold to hot tumors, and you can also see the induction of antibodies and T cells in the periphery. This is also associated in combination with checkpoints in a small trial that was published by Chara's group and now is expanded with AstraZeneca in clearances. So here looking at a person with a head and neck cancer positive, and there's clearance in the presence of the vaccine, after the vaccine plus checkpoint. Same here and same here. And overall, checkpoint by itself gives about a 3 or 4% clearance rates, but here in this small study, it was about 50%. So these are now being studied in more detail in expanded studies. So if we move to now with documentation of CTLs in multiple trials, I've skipped some others, and seroconversion, as I talked about there, we're now talking about infectious disease, and I'm going to take you through a few examples. So one is a paper that was just published online, and that is from the HVTN in collaboration with our DNA vaccine, studied in normal healthies, comparing different formulations and deliveries, IM and ID. And what I'm showing is a comparison of CTL responses compared to different groups. And you can see the the HVTN-098 study, which is a vaccine we studied here, which is a combination of NVGAG and PAL. Or the previous study gives very consistent CTL and CD4 T cells, as well as about 96+% seroconversion and antibody rates, and as well as about 75% of these volunteers have tier 1 newts in the ID delivery. And so this allows you a comparison with multiple other protocols that the HVTN has studied, and the article goes into some detail about, and you can see by the title, the thoughts of the study group of HVTN. So, and these are some of the emerging diseases we had been sort of moving forward with over time. We moved Ebola forward, and that was published in JCI, 100% response rate post-dose 2, led by Pablo Tebas. A MERS vaccine, I'll show you some more data on that because that was our first coronavirus that was published by Maura Gerard and Lancet just last year, and we have a follow-up study. And then Zika was about 6.5 months and had robust immunology and was considered one of the top 10 clinical trials of achievements of 2018, shown here. So this is that study. I'll go through it very quickly. Basically, it's ID delivery, 2 different dose groups, and 1 milligram and 2 milligrams. And you can see basically everyone seroconverts. There's high titered antibodies. They all make, almost all of them make neutralizing antibodies. This is pre, you can see the virus infection green, post vaccine. no infection. And in addition, if you take patient sera, you transfer it to mice, and you challenge them with these are susceptible mice with lethal dose of Zika, they succumb. And so if you take pre-vaccinated volunteer sera, none of the mice survive, but post-vaccination, all of the patients studied have protective antibodies in this model, in this passive transfer to mice. So we move to MERS now. MERS was our first foray. We had done a lot of different work in this model and focused on the spike antigen. Model is shown here of that construct, that predicted expression of that construct. And basically the design is shown here. It's a full-length spike and was created by looking at the sequences, designing them on the computer, putting them into the preferred plasmid that Inovio has developed, and delivering it again into the animals. And then eventually advancing to the clinic. So I'm summarizing a lot of data here. Basically, I'm skipping the small animal studies. There have been several papers, but we developed high titer antibodies in the non-human primates. We had neutralizing antibodies. All of the animals developed T-cell responses that could be quite robust. There was no real significant dose response in the monkeys, even they were pretty, no p-value to that. And it was very dramatic at preventing disease, as shown here. This is a control animal. You can see the inflammation and you can see the disease. And the vaccinated groups, high or low dose, were highly protected. They were protected several logs of viral load. They were protected from virus and dissemination. Remember, this is a 4-site high-dose installation challenge model, short-term. And we've had several more challenges, both with IM and ID approaches, all showing a very high protective ability in this model without any enhancement of disease. This is the study I mentioned about Kavon moving to the clinic. It was, I'm going to show you some features of it. Antibodies were induced in all 3 dose groups, 670 micrograms up to 6 milligrams. There was not really a clear dose response. The antibody responses were equivalent to those in convalescent patients, as shown here, and the antibody responses lasted quite long-term, as shown, followed out here, 60 weeks in most people in the study. In addition, this induced CTLs, T cells across the board, in, as shown here. There was, again, not a clear dose response with a lot of variability in the T cell assays. However, overall, they had higher T cells induced than were induced in convalescent patients in different studies shown here. This is the convalescent patient responses in the lower right, and that's to spike, and that's the GSL vaccine T cell responses summarized. So with this background, CEPI, the Coalition for Epidemic Preparedness Innovations, which I'm sure most of our listeners are familiar with, supported expanded studies of MERS, as well as now one with LASA for the team. And then when we started seeing the first cases in December and then towards January of SARS-CoV-2, they reached out and picked initially 3 groups and now expanded that to another 5 to fund. And the first 3 teams were GSK with Queensland, Inovio, and the Wistar team, and Moderna and NIH. And that was announced in Davos in January. And so this led to our, of course, expansion of this program. And this is kind of the way that program was rapidly assembled by the team between Inovio and the Wistar group first, and then expanding to UPenn. And so a lot of construction design was done by Emma, Dan, Emma, of course, for assay development, Dan and Ami, and Carr. And then, of course, at Inovio, enormous number of people, Kate, Trevor, and of course, the leadership of Laurent Jumeau. And so the constructs were advanced through multiple animal models, mice, As you can see here, by day 7, you have T cells in the periphery, and they develop very good antibodies from single shot, for example, guinea pigs as well, and robust titers to RBD in the animals as well. And so a host of assays were put together. We mapped T cell responses in the mouse study, showing that multiple epitopes could be induced targeting the spike antigen, as shown here in this study that was now just reported in Nature Communications. And in addition, neutralizing antibodies using live virus or pseudotype assays were developed that showed that mice could induce quite consistent neutralizing antibodies. And in guinea pigs as well, in different studies at different doses, you could have quite consistent neutralizing antibodies induced. In addition, Dan's group and Stephanie's group at Inovio both developed ACE2 inhibiting assays, and Dan's group has really expanded those ACE2 blocking, and the immune responses produced in all these animal models do compete with ACE2 blocking as shown here, supporting again the induction of RBD antibodies and neutralizing antibodies by different assays. In addition, we've looked at, we've been studying non-human primates and rabbits and among others. And again, T cells are induced that can be seen quite quickly, and high titered antibodies, 10 to the 5th to 10 to the 6th titer are induced in the non-human primates. And of course, these significantly overlap towards the higher end of for example, convalescent patient sera has been reported by the recent Michelle Nussensweek paper. So with that as sort of the preclinical, some of the preclinical background, Inovio 4800 was, of course, developed as a product by Inovio, manufacturing, et cetera, et cetera. Thousands of doses were created to support the initial studies, which can be done very rapidly with DNA. And they were allowed to proceed in about 10 weeks since the start of the program. So that was a quite rapid advancement. And that entered the clinic, and it was allowed to proceed the week of April 3rd. It took 3 weeks to fully, just under 4 weeks to fully enroll the first cohort of 40 people, 2 different dose groups, and that's done led by Pablo Tebas, shown here in his clinical duties treating COVID patients. Pablo has really been a champion of leading many different first-in-human studies for a variety of groups, including ours in the NIH, and a pioneer in DNA, and of course, John Irwin at Kansas City. In addition, Inovio's partnership with IVI and the Korean Institute of Health was announced, and this was funded and announced that it would advance the clinical trials. Today, Inovio, of course, and IVI announced that that trial has now been allowed to proceed, and so the phase 1 and phase 2 will be recruiting patients and rapidly sort of expand to different age groups over the next few weeks. And so an enormous amount of people contributed to this work. And so I know I've highlighted some of them as we went along. I think the clinical regulatory and development teams really at Inovio have been extraordinary. All the collaborators, all the funders, the Department of Defense, of course, Bill and Melinda Gates, SIPI, myology, et cetera, et cetera, et cetera, AdVaccine in China, which also is expanding the clinical programs. And many of the other collaborators are listed here. And of course, UPenn, enormous support. And of course, the Wistar Institute has been just quite a fantastic supporter of this. And so this coalition keeps growing of groups that are coming in to support the program and expand the trials. And so we hope to see how that goes over the next few months. I want to point out there's many other groups that contributed to the work I presented, so I'm going to that slide as well here. And that was a lab meeting before the new guidelines. So So that's what the purpose, one of the purposes of all of our work on the call today is to get us back to hugs, handshakes, and group lunches, and as one community. So thank you all for allowing me to present. I'm happy to answer questions.
Dr. Weiner, thank you so much. That was fantastic. We're getting a lot of questions. I'm collating them now. I'm just going to share my screen so we've got your title slide back up. Okay. So we're getting some questions about different aspects of the DNA technology. Can you go into a little more detail about the nanoparticle vaccines via adaptive electroporation? This was described as a very exciting tool. People had some questions about that.
Well, thank you for that question. I didn't actually have time to go into that today, but we have been pretty interested over the last, so once we started having the consistency of immune response in the clinic, which really changed, I would say, about 9 years ago, with the combination of the new approach to the device, some of the, and now even more advanced parameters developed by Inovio, with the better formulations and the better expressing cassettes and the higher concentrated formulations, we started to see much better and much more consistent delivery and simpler delivery. We could use a lot less. And we started to think about other applications because we noticed that, as I showed on some of the early slides, the green fluorescent protein deliveries, for example, the whole area was lit up. That suggested that that local area, which is really millions and millions and millions of cells, might be a local place for protein production. And so one of the examples we use of that is the delivery of biologics, and we've published, I think, about 11 different papers on different forms of antibodies and bispecifics targeting different disease models as examples, moving up through our recent publication this year in non-human primates with delivery of 16 different HIV DNA-encoded MABs. And there's a lot of engineering and design in there for non-natural antibodies as well. But that also led to, well, what other shapes can we do? And we had collaborations with different groups, including Bill Schief and of course at our own institute, Dan Culp, to try to incorporate different forms. And we've made many, many different forms of nanoparticles now we've been using, we've been exploring, and those nanoparticles induce antibodies and CTLs. They have a unique property. It was reported in the first paper, which was just published in Science Advances with the group, of course, and Inovio's formulations and concentrations. And so it lowers the amount of dose we need, but it also is very interesting because of the ability to drive CTLs, which we've shown with DNA before. But in this case, it's something that recombinant protein does not do very well. And so it's an early approach. It's an interesting approach, and we're going to be developing a lot more of it. And, we look forward to any advice or help from or interest from other individuals talking about it, because I think this whole idea of moving to this kind of non-live platform that has a lot of consistency of delivery, a lot of advantages in multiple populations. For example, cancer populations where we might want immune therapy that is not capable of spreading. There's a lot of advantages. And so we'll be exploring more of that.
Great. And can you add a little bit more? Can you discuss the immunogen designs that you're considering? Do you add additional sequences to stimulate TLRs and other innate immune receptors?
So that is a very important question. What are our genetic adjuvants? So we have published a course in the clinic, in particular on some of the advantages of using a very IL-12 as a plasmid, as a mixture of basically a low fraction of the whole volume, because we're really delivering in a very small volume. And we're delivering these in 100 lambda volumes. I mean, these are very concentrated formulations. That's the whole footprint in the patient's arm. And so we're delivering very low concentrations. And so IL-12 in, for example, the HVTN publication for HIV and in the Ebola publication clearly shows that you can have an adjuvant effect with that. However, with some of the formulations, we can just go straight with concentrated DNA and get a very robust response. In fact, the coronavirus work I showed here is without any genetic adjuvant. As far as the adjuvants that the DNA induces, it's much different than RNA, for example, which induces a plethora of different pathways. At least I think Drew Weissman's group has published 24 or so different PAMPs or DAMPs, which have a lot of different components to it. DNA is of course quite ubiquitously expressed in our bodies due to the fact that we're colonized by 1 trillion bacteria who shed plasmid. And so we don't make enormous responses in response to plasmid as we originally were thinking a few years ago, but we've seen that it's sort of a mild inflammation at best. Otherwise, we would have a lot of trouble with all those microorganisms shedding so much DNA. When we eat lunch, we have grams of DNA. And so it can stimulate TOL9, we're quite familiar with that, but it's relatively weak at hitting a lot of other pathways. There is TANK1 binding kinase that's stimulated as well, but it's relatively minor compared to some of the other adjuvants. But it is enough, clearly, with the combination of local EP to drive an immune response, and not an inflammatory immune response, but an interferon gamma-based response that tends to drive antibodies and CTLs.
Okay. That sort of alludes to a couple of other questions we received about the immunogenicity of these vaccines. They were asking about, the suggestion that sterilizing immunity is difficult to acquire, although we do observe neutralizing antibodies and protection from severe illness, similar to the adenovirus-based vaccines. Are there things, you mentioned earlier, the adjuvants, are there other things you could consider to improve the immunogenicity of these vaccines?
I think so I want to sort of cut her off. I want to answer that question two different ways. First of all, if we compare the data in the HVTN study, it will be quite clear the antibodies induced by the DNA, these are not the older DNAs people are used to. They're pretty close to some of our best vaccine-inducing vaccines. And they're very consistent, and they can also drive antibodies and T cells even in older adults, which we're now been studying in cancer. And I would challenge the groups to show as good immunogenicity with multiple other vectors in that cancer setting, quite honestly. And I think, so I think we have to stop thinking of them the way we used to think about them.
Sure.
And really see what they're generating now. I think also other groups haven't really tried to deliver them exactly this way, built this way. So the antibody responses are induced and CTLs. And so I think that we can also do things to further engineer them. And for example, we do include IL-12 as a plasmid and some other, we published on other genetic adjuvants. But in the case of COVID, we do have several papers coming out talking about convalescent patients and their responses now. And what you can see is that somewhere between 17 and 20% of people really have almost no neutralizing antibodies, and yet they get better. And so the whole question of what the response is that one needs, and also I think is going to be determined empirically here more than us knowing that answer. Because actually the other issue we're seeing is that people who get the sickest frequently have the highest concentration of neutralizing antibodies. And so I think there's a lot to learn about this disease and a lot more for us to study and try to address those questions.
Absolutely. Yeah, this relates to a question we received about the most vulnerable populations. As you know, there's growing concern that CoV-2 vaccines may not work as well in those most vulnerable. So the elderly and those living in LMICs, which is of particular interest to the Human Vaccines Project, where immune senescence and immune dysregulation occur. Is there any experience from Inovio with immunogenicity of DNA vaccines in either the elderly or those living in LMICs?
So we have done, I've shown some of the studies, the cancer program is primarily in older individuals, right? That is, the higher risk group there. And so we have a lot of women and men, and also we even have, of course, people who are post-chemo treatment. In fact, the GBM study that I didn't talk about that was presented at ASCO just a week ago, I believe, by David Reardon from Harvard, showed that 84% of people do not progress, about 80-84%, with a several dose regimen plus of the vaccine plus checkpoint inhibitor and induction of T cells in most people in the periphery. And that's quite a difficult group to generate antibodies and T cells in. And so I think, I'm not saying we're satisfied with any of that, but I think that's an interesting start. You know, the traditional survival rate at that point is 40 to 60%, and so that's an interesting outcome as well. And so I think we have to follow that more, but I think the ability to, though we've been focusing on that age group as well. We also have a flu study where I think we've gone up to people over 80 and seen antibodies and T cells in that group as well. I don't have numbers from that group yet, so I can't really say definitively, only anecdotally. So I think we'll have more work to do there, but I do think you're raising a very important issue in vaccine responses across populations. We think this non-live approach and non-spreading might have advantages for safety and immunogenicity in such a population.
Absolutely, yeah. Okay, let's see. We're receiving some questions about the details of the insertions. Can you please describe some more detail on the spike insert? Is it stabilized? Is it secreted or transmembrane? And do you have protection data from NHP yet?
Thank you for that question. So the spike, I mean, we published the structure of both the MERS and the SARS-CoV-2. It's full-length spike. As you know, the RBD is kind of a sampling RBD, and the effect of different stabilizations are still being investigated. It's a sort of more fluid situation. And we built it on our previous MERS experience, which is a sort of partially stabilized, but not really as dramatic as some other groups. And I understand why people want to make those different constructs. Those could have very important outcomes. However, you know, in non-human primates and small animals and in humans, we develop neutralizing antibodies and CTLs, and with very good consistency. And I think so. That's basically the background. We have a novel synthetic leader that was put on there to increase the efficiency, and it is a full-length spike.
Okay, great. And just a question going back a bit, how do the SARS-2 specific neutralizing antibody levels elicited by the vaccine compare to the SARS-2 specific neutralizing antibody levels in COVID-19 convalescent sera? Has that been compared?
So convalescent patient sera, so okay, let's talk about that a little bit. I mean, there's been some recent papers. You saw Michelle Nelson's week paper, which is now out. It said about 17% of people don't have any neutralizing antibodies. And then the next 78%, I believe, have a mean of 212 with up to 1,000. That means more than 50% have below 100 titers, and almost 20% have close to no neutralizing titers. And there's only 2 or 3 people in that whole study that had high neutralizing antibodies. And as I mentioned, the highest neutralizing antibodies we're hearing and seeing reports tend to be in the sickest people. So there's 2 questions to that. One is, it's clear that we should be treading lightly on what we think are correlates or not. And we still believe we want a collection. In our MERS challenges, it was unclear. It was very hard to prove how, whether it was neutralizing antibodies or CTLs that were protective in those animals. They're small groups, of course. And I think the fact that 17%, and we're hearing more people recover without having neutralizing antibodies, that's not a trivial percentage. That's actually, that suggests that neutralizing antibodies alone not maybe the only way to control this infection. And I think that's a good thing, because it means that many of the vaccines people are talking about, which induce, are all interesting and might be worth studying. You know, we're very biased as a group. We come because many of us have worked on HIV. And for HIV, we want pounds of neutralizing antibodies. We want truckloads. We want to throw that on. But this, I think we're gonna take a deep breath and just watch how the data comes out and follow that. I do want to say another group, of course, Dan Brook's elegant paper showed that a DNA vaccine was protective without enhancement in non-human primate study. Our studies are behind those. However, you know, we have, I think, 7 different groups in MERS challenges with protection without enhancement. It's basically the same model. We published that before they adapted it, and Heinz group adapted it. And of course, I would say the BioQuals is even a little more slightly different in that some of the features, but it still has features of those installation challenge models. So I think we'll look forward to seeing how the data comes out.
Yeah, it should be interesting. Beyond the neutralizing antibodies, have the DNA vaccines induced other functional antibody characteristics, such as ADCC and other system serology assays?
Yeah, that's a great question. You know, we have published quite a bit on different assays that can do ADCC. We've shown actually in cancer they can kill targets very efficiently. In fact, in our in the paper we have talking about with the HVTN, you know, Guido Ferrari's lab, which is the core lab at Duke, published the functional immunity of the antibodies. So yes, they do all that. I mean, those are all been exciting things to see develop over the last few years as the technology has gotten more consistent and induced better responses.
Okay, great. You know, I was really impressed before when you presented the different Zika and Ebola timelines to the clinical trials. The Zika timeline, if I remember correctly, was about 6.5 months. It seems really fast, and it's really impressive how quickly that got moving. Can you talk a little bit about, you know, the advantages and the challenges of DNA vaccines in terms of getting from pre-clinic to the clinic to manufacturing? We're getting a lot of questions about manufacturing and scale up and what the timeline would look like for these types of candidates.
So the Zika was, you know, that's a whole program. So that's not like from making a construct. I want to point that out. So that 6.5 months developed the reagents, and that was not this.
Oh, okay.
You know, yeah. So that is a very short period of time. I want to point out for the whole program. So I agree with you. Yeah. And basically, one of the things we've seen with the last few thousand people in these studies I mentioned, because that's what it's up to, is a very consistent manufacturing, production, and formulation and delivery. And we're starting to get more and more and more data in these different studies showing a pattern of the antibodies coming up, like I showed with the Zika, and the antibodies coming up with, I didn't show with the Ebola, just the same way in a sort of somewhat consistent pattern. Of course, they're not 100%, but they're completely different patterns that we used to get. It used to be, you know, much less efficient. Now it's quite efficient, and then of course the T cells come up as well. I showed that rapidly with going through the different animal models, and so DNA can be has aspects of a platform, and that platform is not really influenced by the backbone of the vector very much, right? It's not like a viral vector, which has positives in that it can drive responses you want, but there's a tailoring to that, which people use, or even the same way that RNA might drive different pathways and help influence the response. DNA is a much, I would say, it doesn't drive as many stimulations in that way. And so basically, but it's consistency of delivery and simplicity, and the consistency of delivering the same amount locally in that specific tissue, I would say, is influencing what we're seeing. But that's quite a bit of hand waving, to be clear. So I think what the outcome is, though, you can, with some high level of frequency, have the comfort that you're going to see responses in people from a limited number of doses with skin delivery, and those are going to come out in a range that should be something like the non-human primates. I mean, of course, non-human primates, I like to say, exaggerate. Mice lie, primates exaggerate, and clinics
Absolutely.
surprise. So, we expect if we see our MERS data, which was quite consistent and very close to 100% response rates, we hopefully will see something similar.
Great. It's really exciting data. Similarly, we're getting questions about the robust CTL response that you're describing. Are you assessing the epitope-specific responses in vaccinees? And if so, which technology are you utilizing? It would be interesting to hear more about the magnitude and quality of the vaccine epitope-specific CD8 T-cell responses induced. And discussing, we talked a little bit about the convalescent sera and people who recovered from COVID-19. It'd be interesting to hear about the comparison there.
Right. Well, so I think we've been publishing quite a bit of CTLs and some of the other models, defining that we have them in the infectious disease models, but really mapping and showing them more in the cancer model. So people can get an idea there looking at those papers. You know, we're studying broadly the responses in this particular trial. We did generate mapping data, which was published just last week in Nature Communications of the mouse studies, and we'll follow along in the clinic. You know, there's a lovely paper that just came out by SETI's group showing using specific assays for following T cells in groups. And so I think there's a lot of new and exciting assays groups are using. Elias Haddad, for example, at Drexel, who we collaborate with, has a bunch of assays as well. And of course, the Inovio team and collaborators has a bunch of assays, and Penn has quite a few exceptional people, such as Mike Bett's lab, who has had quite a few papers finding T cells. So I think we'll see how these groups, what they learn and find.
Great. Okay, so we have just a few minutes left. Are there any final words you want to comment on next steps or what you're most excited about to see move forward? And specifically to COVID-19, you could comment on next steps. That would be great.
So I think Inovio has publicly announced they'll be sharing data towards the end of June, some preliminary fashion. So we're very much looking forward to that and seeing how that data looks in the studies. You know, in all of these skin studies, we've seen very, very, very well tolerability and no systemic or any type of issues like that. And so we're hopeful that that's what we're going to see here. And then in addition, of course, we're looking forward to seeing the immune response. In addition, of course, Inovio has announced that we've been moving into an FC trial this summer. So we look forward to make that. The pivot has already started. And so we'll look forward to seeing that. And of course, the announcement that IVI is now entering the clinic in Korea with expanded studies also will provide a lot more data at a different location that should be of great interest, and that's of excitement to us. So I think there's a lot of different things that could be very interesting here that we'll learn from.
Absolutely. It's a really exciting time, and thank you to you and the teams and all the people who contributed to this work. It's so impressive, and it should be an exciting summer, I think.
Yeah, we'll look forward to seeing how everybody does, and you know, it's us against the virus. So let's go, guys.
Absolutely. All right. Well, thank you so much, Dr. Weiner. We really appreciate you sharing your insight and knowledge on COVID-19 with us today. And I want to thank all the attendees for your participation. You know, we realize everyone is especially busy and it's been a challenging time. So thank you so much for joining us and sending your questions today. Our next speaker, I invite you in 2 weeks' time to join us on the next Global COVID Lab meeting, which is scheduled on June 18th at 10 a.m. Eastern Daylight Time. Our next speaker is Dr. Eleanor Fish from the University of Toronto, who will present evidence on the antiviral and immunomodulatory effects of interferon alpha, focusing on her clinical studies with SARS-CoV-1 and most recently SARS-CoV-2, COVID-19. If you're interested in more research on COVID-19, you can sign up for our HVP COVID report. In this newsletter, we present insights from experts all around the globe and highlight the latest scientific articles and data. And lastly, please feel free to visit us on our website and follow us on LinkedIn. We're going to be uploading a recording of this webinar there as well. And we really appreciate your time. Have a great day, stay safe, and we hope you'll join us on the next Global COVID Lab meeting. Thanks so much. Have a great day, Dr. Weiner.
Thank you. Thank you again for the ability to present. It's wonderful. You're a fantastic host. Thank you.
Oh, I appreciate it so much. We really appreciate your time. Thank you so much.